All right. Hello, everybody. My name is Danielle Brown. I am the general manager for the Reno site of Charles River. Thank you for letting me come talk to you virtually today. I usually prefer to come in person, but this year we had some travel restrictions from my company, so I had to change at the last minute. So thank you for accommodating, and I hope next year I'll be back in person. So I want to talk to you a little bit about the drug development process. And what that means and why it's so complex, why it's so expensive, why it takes so long, et cetera. So first, a little bit about me. I want to give you a little introduction. I told you that my role right now is general manager for Charles River Laboratories, but I wasn't always in a management role. I actually, my background is actually in science. So I went to college for animal science and then went on to veterinary school. I specialized in pathology, which basically means studying the disease, underlying disease mechanisms. So, for example, if you take your dog to your veterinarian and he has a lump and you're trying to see, is it cancerous? Is it something else? And they may take a sample of it and send it off for a pathologist to look at. That's what pathologists do. Pathologists also work in the drug development industry, mainly making sure that medicines are safe before they go into humans. And that's really the path that I took. So I went into a contract research organization that does work for preclinical or non-clinical testing. So any kind of drug testing before the medicines are put into humans for clinical trials. Before medicines are given to people, there's a long list of tests that need to be done to make sure they're safe and effective. And that's really what my role was in science. I ended up managing a small group of people, a small group of scientists, and went up through the management side of things, mainly because I really like working with people. I like making a positive difference for all the employees. And that's really kind of that led me on this new path of management. And I went back and got my MBA online while I was working, finished that up in 2022. And so now I'm really doing management and leadership full time. However, I work for a very scientific organization, so I still use a lot of my scientific background every day when I'm counseling clients and working with our scientists here. So the drug development process is quite long and complex. This slide makes it look very linear, and I'll show one on the next slide that kind of gives a real idea of what it's like. But from the early basic research of finding a target to make a medicine for, through FDA approval, it takes about 10 years and costs about over $2 billion to make one medicine. Less than 12% of candidates that go into clinical trials in humans are even approved by the FDA. And there's a lot of testing that needs to be done before they even can go into humans. And that's really where I come in. So Charles River is a contract research organization, meaning we partner with drug companies all over the world to help them develop new medicines for mainly humans, but also animals. And over the past 5 years, we worked on over 80% of all the drugs approved by the FDA. So we really get our hands on a lot of different things. We're a large company. We have over 150 sites spread across more than 20 countries. And we have over 21,000 employees. So we have about 700 employees here at the Reno site, but we have a lot of sites across the world. Each year we run studies for more than 1,000 different medications. And that process really begins with knowledge gained from basic research on how disease pathways affect the body. Drug discovery, which is that early basic research path, involves creating or discovering a drug that works on a certain target in a body to treat a particular disease. So for example, there may be a disease with an unmet need. Scientists, chemists, biologists will come together and try to figure out, how do we target this disease and treat this unmet need effectively? Often over 10,000 compounds are screened during this really early research process in order to identify a few that may be worthwhile to go through preclinical testing and then into clinical trials. This early part of basic research and drug discovery takes about 1 to 3 years. We also at Charles River supply a lot of the animals that are used in research, mainly rats and mice. And we also provide some immune cells, plasma, and human donor blood to help with this early research process. A couple hundred medicines may make it through this process and enter into what we call non-clinical development or safety testing. So once it's really determined whether a medicine may effectively hit a target to treat a disease, it's important to make sure that that medicine is safe before putting it into humans. And that's really where our site in Reno mainly works on. This non-clinical development or safety assessment business is what we call it. We provide both animal testing and laboratory testing to evaluate the safety of medicines before going to humans. This process usually, again, takes about 1 to 3 years, same as the early discovery process. And then usually just a very few of these medicines are safe enough to then go into phase 1 human clinical trials. The clinical development stage is the longest stage and the most expensive stage of drug development. Takes about 6 to 7 years. And like I said, only 12% of medicines that enter this phase actually make it all the way through. I'll talk a little bit more in detail about these phases and the number of subphases in clinical development. And usually only one out of those maybe 10,000 that are originally screened will actually make it through FDA approval and review. And then it can be manufactured for commercial use and doctors can start prescribing it to their patients. However, the process doesn't even stop there. The FDA still pays attention even after the drug is approved to make sure there aren't any rare side effects or things that are happening to patients taking the medicine. Things can be pulled off the shelves at any time or put on hold at any time. So I showed that kind of linear process. This is really what it looks like. There's a lot of there's a lot of iterations, basically a lot of going back and forth between all the phases, especially between a lot of the, what we call laboratory sciences or in vitro part of the drug development where we're doing testing, maybe genetic screening or testing on cells or blood. And that's important in the clinic as well. We may find a certain blood test that comes up positive, or maybe a biomarker in the blood that's higher in patients when there's a toxicity or something's going on, and we make sure we really evaluate that clinically as well. So I mentioned in one of the previous slides that we've worked on 80% of medicines approved by the FDA. The FDA is also known as the Food and Drug Administration. That's the full name. It's actually the oldest federal agency. So they were established in 1906, so over 100 years ago. They're part of what's called the Department of Health and Human Services, and that's actually part of the executive branch of government. So the head of the FDA is actually appointed by the president. The FDA has a lot of different sub areas, and the control of the FDA is over more than just medicines, but also food. So human medicines and animal medicines, all the foods we eat, cosmetics, vaccines, and medical devices, which can be anything from a simple thermometer to a joint replacement or a pacemaker or anything like that. So there's a lot of different areas of the FDA. Their control is very broad. And really what their job is to enforce the Food, Drug, and Cosmetics Act, which is a regulation that was passed a long time ago in 1938. Has gone through a lot of additions and iterations, but that's their job. So everything that is developed for medicines for humans, for animals, any foods that we're eating, they all have to go through the FDA for approval before being released. The FDA is accountable to many groups, so they're accountable to the president because that's who appoints the head of the FDA. They're also accountable to the other branches of government, so the Congress and also the courts. A lot of regulations and laws passed by the courts and Congress are related to the Food, Drug, and Cosmetics Act, and the FDA has to enforce those regulations. They're also really accountable to the press. So there's a lot of media coverage about the FDA, especially during times like COVID when we had, the vaccines were being approved, therapies were being approved. There was a lot of scrutiny on the FDA, making sure that everything went through rigorous safety testing before being released, and also making sure we had what we needed to be able to combat that pandemic. And most importantly, on the right, there are the FDA is accountable to all of us as people. So their main job is to make sure that everything that comes out and is marketed to humans is safe. As I said, the earliest part of the drug development process is basic research and discovery. There are a lot of things you may have learned in your studies and college, high school, and even before that, that are really applicable to this part of the drug development process. It's incredibly scientific. Chemists are involved in designing and making new medicines, studying the properties of the medicines, and really optimizing the structure of the drug to make sure it's very effective, it has the least side effects as possible, and that it's also stable in the body. So think about pills you take. If you have to take something 3 times a day, it's a pain. If you had to take it even more than that, you probably would forget. So part of what we look for in these medicines is how long is their half-life, or how long can they survive in the body and be effective, so that we can reduce the number of times that humans have to take the medicine, because we want to make sure there's compliance. So everything you've learned really in chemistry about chemical and physical properties of matter, chemical reactions, types of bonds, etc., are really important building blocks to how chemistry is used in developing new drugs. And this is really called the target discovery or validation. So chemists will look at what is a target in the body that we can create a medicine to fight against, so that we can fight against that disease. What happens next is a large library of potential candidates are screened. A lot of that involves, huge libraries on a computer, screening and getting hits of what types of compounds or chemical structures could hit a target that we are interested in. And then looking for leads. So doing some testing on those targets that are hit and understanding what are some compounds that look like they'll be effective, look like they're going to be stable, and most importantly, are going to be safe and aren't going to have a lot of effects on other targets that we're not interested in hitting. There's a lot of other scientific disciplines that are involved in this process too. So molecular biology, a lot of the new medicines coming out are really targeted towards certain genetic diseases. So we have some new therapies, they're called gene therapies. They're very specific. They can be as specific as a treatment for one person and their specific mutation. And this is really important from a molecular biology standpoint, to know how to do genetic sequencing, how to find where that mutation is, how to create a medicine that will target that mutation and help cure that patient. Cell biologists work with the chemists in tandem to understand the targets we're looking at, where they are in the cell. Are they on the surface of the cell? Are they inside the cell? What do we need to do to get to those targets? What are the pathways we're looking at? So again, a very basic science that you have already started learning in your studies. And then another area is immunology. So vaccines are always going to be important for different diseases. And then cancer research is an area that more recently has really developed related to the field of immunology. So for example, a lot of new therapies for cancer, instead of being more broad therapies that may affect any rapidly dividing cells. We know people that take chemotherapy have a lot of side effects, right? Their hair falls out, they have GI effects, neurologic effects. We want to find medicines that are more targeted for the cancer and not so much for the normal cells. So what a lot of scientists have started doing is developing therapies that use the immune system to fight the cancer. And these are called immuno-oncology drugs. So they equip the immune system to be able to go in and actually fight the cancer cells specifically and have many less side effects than what our traditional chemotherapies have. So this really shows how, basic biology, basic chemistry are the building blocks, but we get more and more sophisticated with the types of therapies we're developing over time as the science becomes more and more sophisticated. Other things that you really need to think about during this process really early on. So think about this is 1 to 3 years, preclinical testing is 1 to 3 years, and then you get into the clinic. But even this early on, when they're first developing a compound, we're thinking about the clinical development plan. How many patients do we need to recruit? Where are we going to run the studies? Those need to be thought about years in advance to get enough enough people to sign up, to understand what where the studies are going to be held, et cetera. The other thing that's already on people's mind in this process, this part of the process, is manufacturing. How are we going to manufacture this medicine? Is it going to be scalable? Are we going to be able to manufacture it in large quantities if it's for a disease where we would need to be able to manufacture in large quantities? So those are things that also help with this kind of lead selection of compounds and optimizing those compounds so that they can be manufactured well, so that they're stable in the body, et cetera. The data from this lead optimization stage, or the stage where the basic science is used to find a couple compounds that are really stable, really effective, can be manufactured, et cetera. All this data is used for some other tests that basically allow us to really pick a candidate that's going to go through the process the best and we think is going to make it into clinical trials. So this includes, like I said, looking at how it will be manufactured. What's the chemistry behind it? Is it stable, or does it break down really easily? Is it stable in the body as well as outside of the body? Sometimes you get pill bottles that are dark because a pill can't be exposed to sunlight. Those are things you have to think about. We also need to look at the chemistry and make sure that that the chemistry is stable, that we can manufacture it, and that we can manufacture it without impurities. And if we have impurities during the manufacturing process, what happens to that medicine? Does it break it down? Does it make it toxic? All of those things. We then start looking at what we call in vivo models, or actual testing in a live species. So an animal, sometimes there's even testing in lower vertebrates or invertebrates. Fish, zebrafish are a big one, or even flies. Starting to look at that, the safety and the efficacy, and then also genetically engineered mice that are animal models of disease. So that can really help us understand if we're hitting the target pathway the right way, is it going to be effective in a human, et cetera. We also look at what's called ADME. So that stands for absorption. So how the drug is absorbed through the body. Do you take it orally and it goes through your GI tract? Does it have to be injected? Distribution, how it gets throughout the body, what tissues it goes to, how quickly it gets there, where does it stay there? Metabolism, how is it broken down? Is it metabolized in the liver and what is it metabolized to? Does it create toxic metabolite compounds when it's broken down in the body? And then how is it excreted? How does it come out of the body? Does it come out through the urine, feces, sweat, anything? So that's something we look at really closely. We call that pharmacokinetics, or PK, which is basically, what the body does to the drug, how the body breaks it down, absorbs it, excretes it, et cetera. We're also going to start looking at toxicology. Toxicology is safety. So you may think of, toxic substances in the environment. It's the same thing. We're just looking at the toxic substances in these drugs when we're making them. So we'll start looking at, different doses. What dose would we want to give to a human? Do we try that dose out in an animal? How high do we want to go with the dose? We look at some safety pharmacology, which is, what does the drug do to certain systems in the body and their function? So the heart, does the medication change the heart rate, the blood pressure, anything like that? Respiratory system, does it change, how the animal's breathing, or respiratory capacity? We also look at genotoxicity. So we want to make sure that any medication is not toxic to the genetic makeup of a cell. Anything that's toxic to the DNA of a cell is pretty much going to be completely shut down at that point. So those are early tests we do to make sure if anything changes the DNA of a cell, it's pretty much killed at that point. So these are a lot of other early tests we do before we say, is it ready to go into this preclinical safety testing in animals in preparation to be given to humans? This next step is preclinical research. So this is where we do a lot of the work here in Reno. And this is really, once a new medicine is developed, it's optimized, we know it's effective, we've seen early indications that it's safe, and this is where we're really proving to the FDA that it's safe. The FDA requires tests to be run to show this, including in vitro laboratory tests where we use chemists, biologists, et cetera, and all different kinds of disciplines across the board, as well as in vivo animal tests. So in vitro is really anything that's done in a test tube, in a petri dish. Well, in vivo means that it's done within a living organism. There's a lot of scientific disciplines that are important during this phase. Like I said, chemistry, continuing to analyze the stability, the formulation of the medicine, its properties. Biology, how does the medicine affect the body, and what does the body do to the medicine? Toxicology, making sure the drug is safe. So we have toxicologists that work on our studies. They kind of are the single point of contact for these studies. Most of them have masters or PhDs in toxicology, and they really run the study. They are the main overseer of the whole study, and they prepare the long report that's sent to the FDA for approval. We also have animal care teams. So the testing that we have to do on animals, it's because we don't have any other way. We're working very hard to find replacements. So we have what's called the 3 R's, which are refinement, replacement, and reduction. So replacement's the first thing we think of. Are there any tests where we don't have to use animals? We found a lot of ways to do that already with some of the other early testing or in vitro tests. If we can't do that, how do we reduce the number of animals we use to make sure that we're getting the data we need from the least number of animals possible? And then refinement. If we can't reduce them any further, how do we make sure that what we're doing has the least amount of pain and suffering possible? So we have a lot of teams that work on this. We have veterinarians on staff. We have veterinary technicians, behavior teams as well. Some of the similar tests that were done in the very early stages are done again during this stage. So looking at the chemistry and the manufacturing of the medicine, looking at the ADME, so the absorption, distribution, metabolism, and excretion, and then doing what we call a GLP toxicology study. So GLP means good laboratory practices. This is what the FDA requires us to do. It's really, all it means is that we have a lot of documentation on our studies, so that when it's submitted to the FDA, they can reconstitute the entire study, know everything was done according to the regulations, and that there was no data falsification or anything like that. All of this goes together in a very, very large package called an investigational new drug application, or IND, and is sent to the FDA for approval before going into humans for clinical trials. So usually it will be submitted around 30 days prior to beginning clinical trials. If the FDA doesn't respond in 30 days, the clinical trials can start. They can still find issues. They can put the medicines on what we call a clinical hold until those issues are figured out. But they usually will submit within 30 days any issues they see. This includes any information on the drug itself, all the information on the toxicology testing, a really thorough plan for what the clinical trials are going to look like, and then also stating that all the clinical trials we run under informed consent and by what's called an institutional review board. So once things go into humans, there's a whole nother step that needs to be taken into account, and that's making sure that everyone that's on the trial has given informed consent, that they know they can leave the trial at any time. So some of you may have been, involved in research studies or clinical trials before, and there's a lot of paperwork that you have to sign. But this is all to make sure that anyone that's involved in these is given their full consent. I said before that clinical research is the longest and most expensive stage for drug development. It takes about 6 to 7 years. Phase 1 is the first phase, and that's a smaller number of individuals, and it's usually healthy volunteers, so people that do not have the disease, up to 100 healthy volunteers, and this is primarily focused on safety. So just like we worked on safety and laboratory tests and some animal tests, this is safety in humans before giving it to patients. If the drug passes this phase 1, it will go into phase 2. Phase 2 includes up to 500 patients that have the disease. And in addition to safety, we're looking at efficacy. These patients have the disease, so we want to see if the medication is effective against the disease. This also helps to understand what is the optimal dose and frequency of dosing to give to humans that have the disease. After this phase, the company that's developing the medicine has to meet with the FDA to talk about any concerns or anything that needs to be addressed before going into the final phase, which is phase 3. Phase 3 is the largest, up to 5,000 patients, usually multiple locations and usually international, so locations around the world, to make sure that there's a diverse population. And these studies are looking for rare reactions. So anything found during any of these phases has to go on the insert for the drug if it's approved. So you've probably seen commercials on TV, or if you take a medicine, there's a large, long paper list and has every side effect that's ever been seen. That's where these come from. They come from these clinical trials. Once the medication is approved by the FDA, it goes into post-market observation. So like I said, the FDA is always watching these drugs. If there's any side effects that are reported that are adverse, it may put it on clinical hold, require additional testing. There may be rare or longer term effects that are reported. Also, if a different population should be given the medication. So for example, if it's a medication that should be given or that the company wants to give to pregnant mothers or children, additional tests will need to be done before that can happen. So those are, we call phase 4 because it's post-market approval. For marketing of the medication, the company will submit what's called a new drug application, or NDA, or what's called a biologics license application, or BLA. It depends on the type of medicine. So most of the medicines you take are pills. We call those small molecules. They're protein medicines. They go through this, what's called an NDA process. Things that have to be injected that have part of a living organism in them. It could be an antibody, it could be even a bacteria, a virus, so any vaccines. Those are called biologics, and they have a separate license process. Basically, these are huge packets of information, even more than the investigational new drug application, that includes all the data from that investigational new drug application, as well as all the information from the clinical trials. They also have to include the label that will be on the drug, all the information that will be on the insert, and everything, all the risks, benefits, everything like that. The FDA usually takes about 10 months to approve a drug after getting the new drug application, and they'll only approve if there's substantive evidence of safety and efficacy. Medicines have changed a lot over time. I alluded a little bit to this earlier. So we think about, the earliest types of medicines, hundreds and thousands of years ago, were natural treatments like herbal remedies or medicines from plants. Drug development started to follow scientific techniques in the late 1800s. The first medicines were actually extracted from plants until the early 1900s. So for example, you've probably taken aspirin for a fever or headache. That actually came from the bark of a willow tree originally, and now it's chemically synthesized. So in the early 1900s, we started chemically synthesizing what we call small molecule drugs, which are pills that we ingest. Penicillin was the first antibiotic that was created, and that actually came from a mold. So again, from something in the natural environment. And this small molecule really refers to the molecular weight of the medicine. So they're simple chemical structure, they're small weight, they can be absorbed through the GI tract, and that's really liquids or pills that you swallow. Kind of within the last several decades, more and more medicines have become what we call biologics, which are medicines produced from living organisms, anything from viruses and bacteria to human cells. Because they contain living organisms, they have very large, complex structure, too large for your GI tract to absorb. So these are always given by injection. And a lot of new medicines are what we call personalized medicines. So this can be personalized to someone's DNA, like I talked about with the genetic diseases and how we treat those, or personalized to fight cancer cells, like I talked about with some of the immuno-oncology drugs, or even newer, what we call cell therapies, where you actually take out the human's T cells, a type of immune cell, you genetically modify them, put them back in the human, and those T cells can then fight leukemia. So we've been able to cure several types of leukemia with these cell therapies. So it's very exciting, but you can imagine these medicines are extremely expensive to make and expensive for people to be able to take them. Some of you may be wondering about the emergency use authorization. So when we went through the COVID-19 pandemic, a lot of the therapies, vaccines, and even things like personal protective equipment like cloth masks or COVID testing all came out pretty quickly. This is because of what's called the Medical Countermeasures Initiative. So this happened, after 9/11. There was a medical countermeasures initiative that came out that said, anything that can diagnose, treat, or prevent life-threatening diseases caused by threats that are either chemical, biological, like COVID-19, radiological, or nuclear, can follow this faster emergency use process. So that's what was done during the COVID pandemic, where we had enough testing to show safety, but maybe not the full battery of FDA testing when things were first released. However, testing continued. The FDA went through full testing and approved all of those therapies and treatments after the fact. So just a little tidbit in case you're wondering. And as far as contract research organizations, there's a lot of different types of contract research organizations. Charles River, we're just one of them. But really, contract research organizations fit in through the entire process. So starting at basic research and discovery of new therapies, to testing for safety, to clinical trials and support, and manufacturing. So there are a lot of different contract research companies. Some focus a little bit more on certain processes. Some may focus more on manufacturing. Some may focus more on clinical trials. Some may be more across the process. And as far as careers in this field, there's a lot of different types of positions, a lot of stages of drug development, a lot of opportunities for even entrepreneurship, through things like mergers and acquisitions, strategy, even starting, new biotech companies. So I put some on here, a lot of scientific positions, of course, early researchers working in the lab, working with the animals, being a study director, but also on the business side, business development and sales, and then a lot of different support staff. So there's a lot of different areas. If any of you are interested in learning more, I'd be happy to talk to you. And I put my email address here. So if you are interested, you can always reach out to me with any questions you have. So thank you for your attention. I'm happy to answer any questions anyone has. I think I don't know if you're talking, Dominic, but you're still on mute, so I can't hear you.
All right, can you hear me?
Yeah, there you go.
Here you are.
Hi, Danielle. That was really good. Thank you very much. One question I had is there's a lot of companies now that are looking at AI technology and using things like the digital twins. I know Recursion and other companies that are doing that. How do you see that impacting the process, the timeline, the cost? And do you think it's going to be successful?
Yeah, I think AI has a large place in this process, especially early on in the drug development, drug discovery and early research side of things. There's a lot that we've already started working on as far as data mining, in silico modeling, and things like that to predict some efficacy and toxicity. It just depends on what data is available to be able to mine, but I think that will really cut down on that early time and cost. That basically refine the medicine so that we can get the better ones that are more going to be safer and more efficacious going through the process from the beginning.
All right, any other questions? Awesome. Well, thank you so much, Danielle Brown, for talking.